All right, everybody, hello and welcome to today's topic, which is qualitative versus quantitative data. All right, so we are inching our way towards, in this class, our data analysis. And what we're really doing here is circling back to something that we talked about very early on in the course. We're talking about different types of data and what they mean for us. So today is a conceptual overview, and then in the coming weeks in our class, what we'll be doing is working with our data, keeping this information in mind. So this pulls us back to our 4 different types of data: nominal, ordinal, interval, and ratio, but talks a little bit more about how we turn that data into information. So let's go ahead and get to it. We're going to start with quantitative data first. So remember that quantitative data, or the word quantitative, refers to numeric values. All right, so quantitating something or quantifying something means that we're adding a numeric value to it. So we're talking about numeric-based data here. So we start off with a general question here. When we have numeric data, what are we looking for? And look, I was covering the signal in our noise. How ironic. But what we're looking for is a signal from the noise, all right? This is a term that we often use. Sometimes this is literal, right? Searching for a signal amongst noise. But this is often a metaphor for finding some difference or relationship amongst a big pile of data. This is often used in things like MRI, looking for a signal or an image out of the noisiness of the brain. So what we're looking for in this case, all right, when we're talking about quantitative data, we're trying to find some values that stand out amongst all of our statistics or all of our qualities of a data set, right? Regardless of all the variation and the differences in our data set, we're looking for some consistent signal from the data. So this is generally a difference between groups. We would analyze this using a t-test or an ANOVA, or a correlation, a co-relationship between two variables. We're looking for some sort of strength of relationship. And this refers back to our choosing the right type of test to use, and we're in the midst of this in our class right now with the exercises we're working on. We have parametric statistics like t-tests and ANOVAs, and non-parametric statistics using categories or ordinal data. That would be like our chi-squared style tests. So this is the idea. The type of data that we have is going to determine how we can test the data and how we're going to try and detect a signal in the noise, or how we're going to detect a relationship amongst data. So analyzing quantitative data comes with its own set of challenges, all right? So quantitative data is often considered superior to qualitative data, and we'll talk a little bit about why. But quantitative data comes with its own set of concerns, a big one being that we have outliers or maybe inexplicable data points in a given data set. So I have an image here of a scatter plot. We practice making scatter plots. If you have a data point that's very far off from the rest of your data, that might be a cause for concern. All right, quantitatively, it does not fit in with the rest of the data. This also can be true when we're looking for other statistics. We can find outliers or anomalies in different modalities like the mean, median, or mode, right, measuring central tendency. We're also interested in spread of data, the skew or the distribution, difference between groups, things like range, variance, and standard deviation. So I hope that you've noticed in this class, we do a lot of practice with visualizing data. We've looked at bar graphs, we've looked at scatter plots, we've looked at line graphs, because we always want to see if something is suspicious about the data, or sus, as the kids say these days. We want to look for anything that's suspect, right? Something that doesn't look like it belongs to our data set. So I think this image does a good job explaining that, right? Something that's very different. This could be an error in data reporting. This could be a sampling error. This person could really be this different from the rest of the group on this measure. But no less, this is something we want to look for when we're working with quantitative data, is abnormality. All right. It's only the, I don't know how many times we've reviewed these same topics, but they're back again, all right? Because when we're talking about quantitative data, what we're talking about is our standard statistical testing, hypothesis testing, using a significance level, using a p-value. So we're here to review it yet again, all right? If we're testing for a relationship, we're testing against a null hypothesis. We're testing against the idea that there is no relationship present. In order to determine that a relationship is present, we need to find a relationship that was unlikely to exist if the null were true. All right, so the significance level is a fixed probability of wrongly rejecting the null hypothesis if it is in fact true. All right, so this is a sort of wordy definition for explaining that 0.05 is considered statistically unlikely to happen due to chance. So if we detect a relationship that exists at an alpha level less than 5%, we assume, right, that the relationship really exists, though we of course have the chance for error. And we'll be talking about type 1 and type 2 errors here momentarily. Should be a review from statistics. Then we have our p-value, right? Our p-value is our significance level. It's the probability that we set as the extreme, more extreme than to be observed by chance alone. So back to the idea that it's unlikely to happen if the null is true, right? Unlikely to happen if the null or no relationship is true. We have a few other terms here. We've talked about each of these briefly, and we'll talk about them briefly again because they do play a role in these ideas that we're discussing here. We have power, statistical power. What power does is it assesses if you have enough data to detect a difference, all right? If you have enough data to detect a difference. We won't run a power analysis in this class, but it's important to know that power is often used before you run or before you collect data, before you run an analysis. Depending on the variation, the spread of your data, sometimes you need more data to detect a difference, all right? So this is back to our larger samples are more likely to detect significant differences. That means that larger samples are more powerful. You're more likely to find differences, statistically significant differences in larger data sets. And we have an analysis set to tell us exactly how large it should be for us to detect those differences. And then effect size, all right? We've talked about effect size once before. What effect size looks at is the magnitude of difference between or in a relationship, but it doesn't use sample size, all right? If we even look at the calculation used, it doesn't use sample size, all right? It uses things like the standard deviation, which is partially a product of sample size. But what it aims to do is remove sample size from remove some of the power of sample size from hypothesis testing. So effect size is often taken in partnership with standard significance testing. All right, so we have some concerns with quantitative research, many of which we've been discussing along the way here. We need a big enough sample size. We need to run the appropriate statistical test. We need to know what type of data we have and what's appropriate to run. We need to know about our likelihood of making a type 1 or a type 2 error. And this is directly related to our significance level or our alpha value. And again, we'll be getting to this in just a moment. We'll be talking about these types of errors. We want to know if our results are generalizable. Generalizability is another topic that we've discussed a few times here together. Generalizable results mean that if you take a sample of individuals and make a statement about them, you can generalize to a larger population. That's what we're always trying to do in statistics and that we're always trying to do in research. We also want to know if our analysis, our results, our methods, if they're reproducible. If they're not reproducible, they don't hold very much merit, right? Because remember, the scientific method is a cycle, it's a circle, and we replicate and we reproduce findings to increase their validity, right, their accuracy. If we find something once, right, think back to our green jelly beans and acne example, right? Maybe you do find that relationship once, maybe you find that it's statistically significant, but maybe it was an error, right? Maybe it was a type 1 error when we detected a relationship that wasn't actually there. So we can fight this by reproducing the studies, right? Doing it again and again and again to see if we have some consistency. And then am I measuring things the right way? All right, that's back to validity. Are we accurately getting at the concept or measure that we say we're measuring? Sometimes it's not as simple as it seems. So these are our concerns. Let's talk about them a little bit more. All right, the issues, why these are concerns. Some things can't be measured or measured accurately. We have a great example of this, one that we usually look to, which is morality. Scientists, of course, are interested in studying morality. We do have an idea of what most people consider to be moral, but it's not objective, right? It's subjective. A lot of morality comes from different religious backgrounds. So morality is a tricky one. We can try to measure it, but we'll have a lot of contest, right? What's moral to one person might not be moral to another. What's moral in one culture might not be moral to another culture. So it's difficult to measure. Other things can't be measured accurately for different reasons, all right? We have a big issue measuring things like sexual assault because we often have individuals who don't want to report the information. You can't measure something if you don't have any data on it, right? So if we don't have a holistic view of a variable or a set of data, we can't measure it accurately, right? We can try, but we have to be aware of our limitations. And here's the big one, all right, with quantitative research. It doesn't tell you why. You can get a correlation between two variables, but it's not going to tell you anything about why that relationship exists. That's where the researcher comes in and can try and interpret it, can try and look to other research to figure out why a phenomena is occurring. But quantitative research, quantitative data itself doesn't tell you why. It just tells you what's out there, right? What the data says. It can be impersonal. We take human behavior and we turn it into numbers, right? We probably lose a little bit of the unique human experience this way. And this is why we'll talk about qualitative data as well. That's the second part of this talk here today. Data can be static. All right, we've talked about this before as well. Research is a snapshot in time. It's a single point in time. Things change, right? Maybe you conduct a study on confidence in individuals and you conduct it on individuals at the college level. Maybe in 5 years, when you're out of college, people are more confident, right? So that research is just a snapshot in time, and this is why time is such a big issue in our experimental designs and why we have cross-sectional and longitudinal designs. And people can tell lies, all right? People can tell you lies in your data, and statistics can be incorrect. Statistics can be manipulative, all right? So we've talked a lot about this. We need to be healthy skeptics, all right? We need to be skeptical when somebody gives us some information based on some data. We should ask ourselves questions like, where did this data come from? What are the motivations of the individuals publishing this data? Was it interpreted in the way that maybe you would have interpreted it? So you've maybe heard this quote before, and it definitely has some merit to it, right? Numbers are persuasive. Those that are outside of the field of statistics or outside of the field of research think that statistics are sort of the answer to everything, right? Well, once we get the data, we have the answer, and that's not quite true. Once we have the data, we might have a better understanding of something, but we rarely have the answer, and it's often interpreted that way. Or presented that way, right? Many companies think marketing research will take some statistics, right? I like beauty products. I think beauty products do a very, I don't know whether to say poor or a good job of this, but they market, right, saying your hair will be 80% silkier, right? If you use this conditioner, this shampoo. That's a bold claim, right? 80% silkier, then what, right? So you want to start to ask these questions, right? When you read statistics that are used to influence you, you should think about why that influence might be there in the first place. And it's often shrouded by numbers, right? It's like, well, the data says, but the data doesn't always say, right? Researchers speak for the data as well. All right, so now we're on to type 1 and type 2 errors, all right? So this is kind of a continuation on our hypothesis testing. This is one of our chief concerns when we're looking at quantitative data or using significance testing, p-value testing. We're still relying on probability, Okay, we're still relying on probability, and remember that probability is imperfect. All right, there can be a 0.01% chance of something happening, very, very low. It can still happen, right? So we see this in our statistical decision making. Sometimes we will reject a null hypothesis when it's actually true. We'll say that there's an effect when there's not. This is known as a false positive, right? A false positive. A type 2 error is the opposite. That's incorrectly retaining a false null hypothesis. So saying that there's no effect when there actually is. This would be a false negative. So I think that a decision table is helpful for this. I have one here. So we have cases when the null is true, when the null is false, and what happens if we accept or reject it. Though remember, the word accept is a sort of naughty word. We like to use like retain the null. Accepting makes it seem like it's true, all right, when we don't know for sure. We just have some evidence to suggest that it's true. So this is pretty wordy, right? There's a lot of kind of room for like, wait, double negatives, falsely rejecting. So I like to think about this in a couple of other ways. I think this is a lovely example. Well, let's move my head. Here we go. So never confuse type 1 and type 2 errors again. Remember the boy who cried wolf? He had both type 1 and type 2 errors in that order. First, everyone believed there was a wolf when there wasn't. Next, they believed there was no wolf when there was, all right? So the wolf here is the effect or the relationship that you're trying to detect. So I think this is a nice way to think about it. And even a better way to think about it, if you've taken my stats class, you know how much I love this particular image. I think it's great. Let me move my head. Okay, because I think pregnancy is a great way to think about this, because we can think about, say, a pregnancy test that's 99.9% effective, all right, but there's still that 0.1% chance of error, right? So let's take a look at how those errors can come to life. A type 1 error, a false positive, would be someone thinking they're pregnant when they're not, all right? So we have a male doctor telling a man, you're pregnant, all right, a false positive. A false negative would be maybe a doctor telling a very pregnant woman that she's not pregnant, right? And this is a little ridiculous, but we can think of this more as our pregnancy tests, right? Peeing on a stick and getting your results. There are false positives, there are false negatives, right? And those are our type 1 and our type 2 errors. So I hope that if you get caught up on these terms that you look to this example. I do think this is the best way to think about it, and a little humorous too. So, all right, with that, we're going to move into qualitative data or qualitative research. So quantitative research or quantitative data refers to numbers, the use of numbers to look at data. Qualitative research, qualitative data is a little bit different. We're looking at the qualities or the explanation of something. So let's talk some kind of examples and some of the ways that we use qualitative data. Qualitative data, unlike quantitative research, tries to get at the how and why things happen. We often do this by asking people, how did this happen? Why do you think this? And in our class, we strategically avoided asking questions like this because, as we'll discuss here, it's very difficult to interpret, all right? Very difficult to interpret because it's subjective, right? And subjective meaning that individuals will give their how or their why. We can't just slap a number on it, right? We need to actually interpret it. A human needs to interpret it to see what it means. So when we're working with qualitative data, we don't need large sample sizes, all right? But we have some issues that we're concerned with. We're concerned with respondents providing inaccurate or false information. We have something called the social desirability effect. This is particularly prominent in things like, what this is people lying, all right, to come off as socially desirable. And most often this happens in cases of saying how much you exercise, saying how healthy you eat, saying how much you recycle or do things that are considered socially desirable behaviors. People will lie to make themselves seem in a better light, even if the data is anonymous and confidential, and we've seen this time and time again. We see this in personality testing. People will rate their personalities generally as more positive than others might rate their personality. So that's something we certainly need to keep an eye on with this type of research. Ethical issues may be more problematic. The researcher is usually closer to participants. Maybe you're having a participant recount a past trauma, right? And you want to understand and write down to have as data, right? You want to have data about how someone, say, processes trauma, but that's very sensitive data, right? That's difficult to obtain from people. We talked a little bit about how when we're talking about things like trauma or suicide, we need to refer those individuals to care. So that becomes an ethical concern. And researcher objectivity may be more difficult to achieve. So again, we're talking a lot about subjectivity and objectivity. To be objective is to be more concrete, and to be subjective is to be more rooted in personal opinion. So we'll talk a little bit more about examples here. Qualitative data requires a lot more interpretation by a researcher, which leaves more room for subjectivity. So let's take an example of this, all right? So this shows us the sort of spectrum from qualitative data to quantitative data. So a qualitative description would be something like, I drink coffee every day. If we go the quantitative route, you might say something like, I drink 4 cups of coffee every day. Or we can measure it another way. How many grams of coffee do you consume every day? Some of you might be using caffeine as a variable, and one thing that's interesting about this is that most people don't know how much caffeine they consume in a day. They can answer you 4 cups of coffee, but people's cups of coffee might be different, right? A double espresso is a lot different than a standard cup of coffee. So this kind of just gets back to the fact that our data is complicated, right? And that we need to describe it the best way that we can. And that qualitative data can be difficult to make more specific, right? If someone says they drink coffee every day, we don't know how much coffee they're drinking every day, just that they are drinking it every day. All right, let's talk about some sources of qualitative data. All right, so we have interviews. This is a very common source of qualitative data. Think in therapy, right? Structured interviews are an important part of, say, diagnosis or understanding a case of a client in the therapeutic setting. So interviews can provide much qualitative data. Focus groups, questionnaires or surveys, you can leave open-ended questions, which again, I advised against for this class because we're talking all about how difficult qualitative data is to understand and to work with. Secondary data is often used as well. Diaries, journals, self-report, written accounts of past events. We have a lot of accounts of, say, people in history explaining other people in history, right? Explaining the qualities of that person that a lot of psychologists have been interested in analyzing. Or direct observations, recording. Sometimes in the therapeutic setting, there will be recordings of clients so that we can qualitatively assess their behavior. An example of this is, there's a study called the Strange Situation. Some of you may have heard of this. The Strange Situation is a strange situation in which you take infants or babies and you put them into an atypical situation where they have their caregiver and a stranger basically entering and exiting a room. So the baby will be with their mother for 90 seconds, and then a stranger will enter the room for 90 seconds, and then the mother will exit the room, leaving the baby alone with the stranger. The stranger will leave the room, leaving the baby alone. Then the stranger will go back into the room and try and console the baby. It's a paradigm. There's all of these different iterations of what'll be done, right? And we want to assess the baby's behavior at any given time. So we observe it, we record it, and what researchers do is go back and code that information. How comfortable was the baby when the mother was in the room? And we code for things like the baby holding on to the mother, the baby staying close to the mother. And we do the same thing for the stranger, right? Are they accepting of the stranger? Will they go up to the stranger? Will they let the stranger pick them up? So we're coding the qualities of the behavior, right? And we often have to videotape it at the time and then go back and meticulously code it. So that's why qualitative data is challenging, right? It doesn't just come out as a number. Like if we ask someone on a scale of 1 through 10, how attached is your baby to you? It doesn't get at the same information, right? It also doesn't get at why that's occurring. So sources of qualitative data are diverse. And then the issues or the concerns are a lot of what we've talked about so far. How do we code the data, right? How do we code for how connected a baby is to its mother in the strange situation? We have to ask if we've captured the situation or the question at hand in a realistic manner, if we're getting the true qualities of a person. You have to ask if you've described the context in sufficient detail. Remember, this is part of research reporting. We want to be very clear, very concise in what we did and how we captured our information. All right, we want to provide lots of detail. Have you managed to see the world through the eyes of your participant? All right, this can be challenging. Again, we're talking about infants, right? Are we correctly assuming the qualities of an infant by watching them on video and coding their behavior with their mother? We could say yes, right? Science agrees for now that that's the best way we can assess that. But we do need to ask ourselves if we're being accurate, right? Are we doing a good job of taking the perspective of our participants? And is our approach flexible? All right, do we have a flexible approach and we're continuing to interpret the qualitative research to the best of our ability? All right, so lastly, our issues, lots of issues and concerns in this particular topic. But it can be subjective. All right, remember our difference between subjectivity and objectivity. It's hard to be objective. It's hard to be matter of fact or concrete because we're using our own interpretations. It can't always be repeated, all right, not in the same way that an experiment can. It can't always be generalizable. Sometimes it's more specific to a smaller set of participants or a smaller set of scenarios, so we don't have as much generalizability. It can't always give you definite answers in the way that quantitative research can. And remember, we never get definite answers, all right? But what we mean here is that we don't get a nice and clean p-value at the end of this, right? We don't get a nice sort of conclusion to interpret very easily a significance test. This is often more exploratory and just explaining what we found. It can be easier to carry out or hide bad qualitative research than bad quantitative research. And this is a big one, right? Because a lot of this is left up to interpretation, and sometimes a researcher might interpret things in such a way that it supports their hypothesis, that they create data that's maybe not really reflective of the qualities that were assessed. So this is a major concern, which is why qualitative data is so difficult to work with and why we generally avoid it in this course, because there's a lot clearer track for analyzing quantitative data than there is for analyzing qualitative data. All right, and with that, that's our overview of quantitative versus qualitative research. This is also our last topic. In terms of our standard PowerPoint delivered topics. The rest of the topics we'll cover in this course will come through demonstrations of data and applying this to data. So hopefully we're looking forward to that. Thank you for hanging in there for this lecture, and I'll see you next time.